First, thank you for the invitation to come talk at the meeting. And as Tanya said, I'm going to talk about some of the capabilities within the EMSL for the functional characterization of plants and microbes. So we've heard a lot over the past day and a half about how plants and microbes and how they interact and how it is important for the ecosystem, so I'm not going to belabor the point except to say that the study of the plant-microbe interactions has a number of implications in the development of new avenues for improved crop health and crop production, as we heard about yesterday, as well as understanding of the terrestrial carbon fluxes that will help climate modelers understand the carbon flow as our climate is changing. We in the EMSL have a number of capabilities to study these interactions, and what I want to try to do is go through these capabilities, and I organized them through scales. So we have capabilities that go from the micrometer scale to the nanometer scale, where we can look at different aspects of these interactions from a gross picture scale to the electron scale. So we have X-ray tomography to look at gross root structures. We have hydrology, where we can look at the flow of the water through the roots into the microbes and back out. We can look at geo and biogeochemistry scales through microscopy, where we can look at the microbes and how they interact with the roots from the spatial organization and how they're interacting with each other. We have organic matter characterization through NMR and mass spec, where we can look at what are the components of the soil, where is the carbon stored, and how is it utilized. We have omics capabilities, which I'm most familiar with, to look at the biomolecules that were within the roots and within the bacteria and the fungi, and then how are they changing as these organisms interact with each other. Lastly, we have computing to go down to the electron scale, and we can model a lot of the interactions between the proteins as well as the electron transfer in these organisms. So starting at the gross scale and looking at some of the imaging capabilities we have in the facility, we have imaging based on mass spec, where we use the mass specs to have an ionization technique that will raster across a surface and liberate ion molecules that are either proteins or metabolites or some other biomolecule that we can then look at in the mass spectrometry and get a spatial idea of where these molecules are. We have electron and ion-based imaging that gives us an idea of how these organisms are associating with, say, root molecules, or how they're spatially oriented within soils. We have NMR imaging that allows us to not only look at structures of these organisms, but then how nutrients flow through the system. If we use, say, a label, we can look at a label moving through a biosystem. We have optical imaging, which again gives us a gross view of the roots and the microbes. And then we have, as I said, imagery construction through our computational capabilities. So just a few illustrations, and I will apologize that we don't have one user project that spans all of these capabilities. We'd like to, but we don't have that. So I'm going to be jumping through a number of different examples, and I will try to orient you into exactly what I'm talking about in exactly the biological system, but it's not going to be the same one. So we had an EMSL Rhizosphere Grand Challenge that was led by Liliana Positalik that was focused on looking at the characterization of the Arabidopsis rhizosphere. And these are images from that Grand Challenge where we can see microbes that heavily populate the root of the Arabidopsis. And if we get a closer look, we can see a number of these microbes. There's different kinds, they're spatially oriented, and we can get an understanding of how these microbes are colonizing these root systems. We can take a closer look and use microscopic probing, where here, again, this is the same, it's not the same Arabidopsis roots, but it is Arabidopsis roots, where the blue label is root epidermal cell wall, the red is the DNA from plant nuclei, the green is a universal bacteria fluorescence, and then the yellow is the green and the red that tells us where the bacteria are. And we can see that the bacteria have colonized the roots in specific places. And as we go closer, we can see how the bacteria is colonizing these root structures and start getting an idea of how they spatially orient on the root to possibly understand how they are interacting with these roots. So It doesn't really tell us a whole lot. It tells us where the bacteria are, but it doesn't tell us a whole lot about what bacteria are there. So our next step is to use fish probes coupled with this capability that will allow us to determine what microbes each one of these little balls are, and get an understanding of how they're populating the root structure and then how they're interacting with each other. So if we can understand which microbes are interacting with each other in a spatial way, we can understand we can start having an understanding of how this whole system works. Okay, I did go. The other thing that we've done is laser scanning confocal microscopy, and this is actually a Ficus project with Sarah Leibus, where we were looking at duckweed. And you can see, using the laser scanning confocal microscopy, where the red is the root cell wall and the green are the bacterial cells, that as this spins, you can start seeing the green start to kind of fill in, and we can see where along the root these microbes are colonizing. And so using this, again, we're getting a visual idea of how the microbes are interacting with the root system. So moving from a gross scale view of the root and the microbes, we want to move into a more finer scale of looking at the ohms, looking at some of the biomolecules within these cells. And so you've heard a lot about metagenomics and metatranscriptomics, which JGI is uniquely and very well, and has shown to be very well suited to do. And we are going to focus on the other ohms, the metaproteomes and the metabolomes, the lipidomes. But it's not easy. So when we normally have a cell sample and we want to do metaproteomics and metabolite profiling, we use what's called an MPlex an MPlex extraction. And what this is it's a methanol chloroform extraction that allows us to separate out the polar metabolites, the proteins within within the interface, and the lipids within the chloroform. Typically this, and I should have had a picture, typically this is very clean and it has a very, very nice protein band, and then you try it on soils and you get this, where we see the debris at the bottom and we see our protein band is extremely thick because there's humics and other stuff in there, and our lipid and our polar metabolite extractions are brown, which means there's an awful lot more contaminants in there. And so we've developed a number of capabilities a number of techniques to be able to get rid of a lot of these contaminants to increase our protein yield and our metabolite yield in these systems. So we feel like we're poised at this point in time to really tackle the proteomes and the metabolomes in these soil systems to get a pretty good coverage of what is represented. So as an illustration on what we can do, I chose the Ficus project that is led by Mary Firestone to look at the relationship between plants and soil microbial communities. And she's working out in Hopland, California, and using an Avena barbata organism. And what they are doing in their lab is they're growing these plants on these rhizotrons and feeding the plants CO2, either labeled or unlabeled, to understand what the relationship between these roots and the soil is. We take the outcome of this and characterize it by LC-MS. And I'm going to talk about the proteomic characterization of this system. So going through using a metagenome that was sequenced by the JGI, we were able to characterize over 68,000 proteins out of our metagenome. Now, we have an unbinned metagenome, which means we don't have, at the moment, organismal identifications of what our proteins are. That means I have 100 citrate synthases and 78 ATP synthases. And so what I did is I took all the proteins and I condensed them down into functional annotations. And what we end up with is about 2,200 proteins that correspond to what I'm going to just call our gross bacterial load in our soils. There is now available a binned metagenome, and we will be looking at which of these proteins belong to which organisms. But just taking a look at these 2,200, which for a typical bacteria has about 4,000 proteins, that's pretty good coverage on our protein load, especially since we condensed all of the hypothetical and conserved hypothetical down to one functional annotation. So we get our identification of our proteins. So we see, where are they? We see nucleotide biosynthesis, we see transcription, we see a lot of energy metabolism. We see the typical proteins we would expect to see. But lists of proteins don't really tell us much. It tells us what the organisms can do, but it doesn't really tell us what they are doing. And it certainly doesn't tell us how these organisms are interacting with the plant. So what we've done, so the way to approach that is to use a stable isotope label. Now, a stable isotope label gives you an opportunity to add something to your system that designates it as T0. And then we can follow that stable isotope label through time and figure out where it went. And in this case, what we've done is we've added a stable isotope label. The first thing we did is we added a stable isotope label directly to soil. Now, Trent Northern did some studies, some metabolomic studies on Avena plant exudates, and he found that one of the major components of the exudate was sugar. So we wanted to add glucose directly to the soil to figure out if we could actually see the proteins take up the label in the soil and then what they were. So after incubation, we analyzed everything by LCM-SMS, and we got, as in the last list, a lot of proteins that weren't unexpected. We see our sugar and our xylose proteins, or transporter proteins, ATP synthase, our glycolytic proteins, our pentose phosphate proteins, and their TCA cycle proteins. These cells are alive, they're functioning. These are just the proteins that we see in, expressed just in our global analysis. Now, I'm going to overlay on here the proteins that we saw that demonstrated a label. And the way we find the label is we look at our mass spectra and we look for a shift in an isotopic pattern indicating that there's heavy carbon in there. So if we overlay our labeled proteins, what we see is a number of these light up as yellow. We see a lot of glycolytic proteins. We see a lot of pentose phosphate proteins, our TCA cycle proteins. We see these PHAs, which are associated with carbon storage and energy. So what this is telling us is that by adding carbon to the soil, we gave it a carbon overload. We gave these microbes the ability to store their carbon and not have to utilize all of it. Interestingly, we did not see, or maybe not interestingly, our cellobiose metabolism and our cellulase metabolism proteins. There's no reason for these organisms to be synthesizing these kind of proteins after the addition of glucose because they have sugar to eat. So they're certainly not going to be degrading anything more complex. So our next step was to take the samples for when the plants were growing in the presence of CO2, track the CO2 into the soils and see what proteins, again, were labeled after when they were exposed to plant exudates. And again, globally, we see some of the same similar proteins. They exist in the soils. But when we overlay this with the things that are labeled, we see very few glycolytic proteins. TCA proteins are pentose phosphate pathway proteins that are synthesized in response to exudates that come from the plant. What we do see, we also don't see any PHAs. So obviously, these cells are not storing carbon. They're not getting enough carbon for them to store. We do see cellobiose metabolism, so it's possible that this is responding not just to plant exudates, but it could be also utilizing root degradation products, and it's breaking them down. We see a nitrate reductase, and we also see this methane phenoltoluene hydroxylase, and that was kind of surprising to me. I didn't know really what that was. So I had these samples run in our NMR facility in a solid state by solid state NMR. And what solid state NMR will tell you is it can break down, it's just a gross level survey of the soil. It's non-destructive, so I can give the person the soil and he gives it right back to me in the same state that I got it, or that he got it. But it tells me what gross level metabolites are there. And so the red lines are week 0, our blue lines are week 12, and we see an increase in our carbonyls. There's a little peak in our aromatics, and there's another peak in our carbohydrates. And we can link these back to specific proteins that we see in our proteomic characterization. Now, interestingly, we see this little peak in our aromatics, and we saw this methane phenoltoluene hydroxylase. And so what we want to do is this gives us some clue as to where to start looking in this system. And we want to maybe figure out what this protein is actually doing and maybe what metabolites it might be utilizing. So again, we're going to be working with Trent to look at some of the metabolomics and start unraveling this whole story. So the other capability we have in EMSL is, or one that's unique to EMSL, is our Fourier transform ion cyclotron resonance mass spectrometry, which I won't say again in this talk. I will refer to it as FTICR. The FTICRs are unique instruments in that they have they have the capacity for high sensitivity and high mass measurement accuracy. So the great thing about metabolomics is we can measure the mass of a metabolite to such a point that we can get a molecular formula from that metabolite. So we've used the FTICR-MS to characterize extracts from soil, where we get our spectra, which can then be plotted in a Van Crevlin diagram, where we plot the ratio of hydrogen to carbon and oxygen to carbon. And as we plot these, we can start understanding the load of lipid to carbohydrate to proteins to amino acids. So the first thing, as in proteomics, we had to do is we had to learn how to extract the metabolites from soils. And Malak Tafali in our group developed a sequential extraction for metabolites from soils for the FTICR. The first step is a water extraction, and the water extraction gets out a lot of the metabolites, including aromatics, tannins, lignans, carbohydrates, and proteins. This is followed by a methanol extraction, which liberates hydrocarbons, lignans, and lipids, followed by an acetonitrile extraction, which extracts aromatics and tannins. And so through the sequential extraction, we're getting a lot of the metabolites that are contained within the soil. And we can analyze each of these fractions on the FTICR individually and get a survey of what is actually in the soils. And so just a demonstration of this, going back to the Arabidopsis rhizosphere Grand Challenge, we looked at the metabolite load in a night cycle and a day cycle from the Arabidopsis, and what you can see is a distinct difference in the number and the type of metabolites that we're getting out of these systems. When we start comparing these, we can start looking at the molecular formulas and then relate it back to either KEGG diagrams or some of the other metabolite data we have to try to figure out exactly what these peaks are. The other thing that we can look at, and again, going smaller, is siderophores. And siderophores are metal binding chelators that are very important for microbial life. And what we're able to do in the EMSL, in the siderophores, is use our ICP-MS as well as our 21 Tesla FTICR. Now, the 21 Tesla FTICR is a very unique capability within the EMSL, and it has the ability to, it has a very high mass measurement accuracy. So as we look at our peaks, we can get, again, it's not only molecular scale resolution on for our identifications, but it also allows us to see the metals. Now, a metal has, for an isotopic pattern for a carbon, a nitrogen, and oxygen, the smallest isotope is the most abundant, and so you get a peak. Your biggest peak in any profile is always the smallest. Metals aren't that way. In metals, most of the most abundant peaks are either the third or the fourth highest in abundance, and so you get small little peaks out in front of the highest, and that tells you whether or not there's a metal. So we can use ICP-MS to find out the fractions that have the metal in it. We can then use FTICR to look at what these are, and just as an illustration on how this works. Here we go. We can look at amphibactin from Vibrio, and we get a typical spectra, and we can zoom in to look at not just our molecular profile of our compound, but it actually shows us the oxidation state of the iron in the organism, or in the molecule. So we can then start identifying what the siderophores are, and if we know what we're looking for, we can look for a specific molecular formula. But if we don't know what we're looking for and we run through this process, we can start identifying unknown siderophores. Okay. So we also have a number of capabilities that do mass spec imaging, and they span, and I've listed them here, spanning, again, something that's going to give you a gross view to an atomic view. And all of these are available within the EMSL, and they're available for a Ficus to request through a Ficus project. But these here will give you molecular scale information, where these will give you atomic scale information. And so using a suite of these imaging capabilities, you're able to get a better view into your system. So some of the things that are on the horizon, we want to be able to do single-cell metabolomics using these imaging capabilities that I just mentioned, where we are actually able to target a single cell, liberate out the metabolites from that single cell, and then use the 21 Tesla FTICR to analyze the metabolites that we liberate. I should also mention the 21 Tesla has an unsurpassed sensitivity of any of the other mass spectrometers, so we're actually able to see very low-level metabolites. We're also looking to do cell-specific proteomics, and this is actually in collaboration with the single-cell sorting here at the JGI, where we want to sort the cells into single cells, and then we've developed these very small reaction chambers. And so this is a chip, and you can see the kind of size, the size of the chip, and it has 395 different wells to do our tryptic digestion. So within these wells, you can put a single cell, do your lysis, do your extraction, and start doing single-cell proteomics. So just to conclude, what EMSL offers is specialized facilities and unique instrumentation. But what we really offer is expert staff that allow you to utilize those capabilities to forward any biological question you have. And as you access the EMSL, you can interact with these people that have spent years and decades not only developing this instrumentation, but utilizing it to answer biological questions. You've heard about this one, but I'll mention it again. The Ficus calls, the letter of intent is due April 3rd. It's a fairly short letter of intent, so feel free to contact anybody in the EMSL or in the JGI if you need information or you need help putting one together. We also have a general call for EMSL proposals due September 1st. That is their shorter, smaller scope proposals that only span a year. Lastly, our 2017 Multi-Omics for Microbiomes meeting is August 1st through 3rd. It's also our EMSL integration meeting. And lastly, I'd like to say happy birthday to the EMSL. We have been in existence for 20 years this year. So we are having a birthday party, and the birthday party is August 3rd through 4th in Richland. Last, thank you for your attention, and I'm more than willing to take any questions.
We have time for a few questions. I think there were questions about imaging earlier. I don't know if they still are here.
I think I talked to them earlier during the break.
Okay, resolved. I have one quick question then. So for the proteomics data, you mentioned, for example, the 22,000 proteins in soil. How do users, respectively, in that case, I guess it's your collaborator, integrate these data with the other data types? So you have genomes from metagenomes from these cells. What are people doing with the data for integration?
That's a really good question, and I think data integration is the challenge that we're all facing right now. Right now, we can map it back onto metagenomes. We can map it back onto pathways that we can fit them into, but there really isn't an automated way to map it back. We can put them into IMG and understand, like, are they along an intron? What pathways are there? But I'd like to look to KBase to help us do that.
Yep. Okay, Great. One more question over here, yeah?
So if I have a mixture of like metal binding protein or metal binding compound, then what would be the best way to characterize which protein or compound contains which metal?
Okay, so the one way to do it is if the metal is covalently bound to the protein, is digest the protein down to peptide. Look for that signature, the signature 2 or 3 peaks in front of the largest peak in our spectra, and then look and see what that, do MSMS and figure out what that peptide is.
If they were not covalently bound.
I'm sorry?
If they were not covalently bound.
If they're non-covalent, what we would do is similar to what we did with the siderophores, and we'd use ICP-MS to fractionate out, get the fractions that contain the metals, and then do a bottom-up proteomics approach where we would look at the, we'd identify the peptides. We'd probably marry that with some top-down proteomics in the FTICR in a soft ionization technique that allows the metal to stay on the protein, and so we can characterize the metal on that protein.
So I still have to separate the mixture of compounds?
Probably,
yeah.
One more question, yeah?
For the single-cell metabolomics and proteomics, how many cells actually do you have to analyze in order to have a complete understanding of the metabolome and proteome?
That's a difficult question right now because these are methods in development right now. So at the moment, we're at thousands to tens of thousands. We want to get down to that stage, and this is the reason the single-cell proteomics is important is because when you do a tryptic digest, you lose so much protein just in cell, your tubes eat your proteins. And so having that microfluidic device that's going to allow us to do that tryptic digestion right there means that we're going to be able to identify more proteins from the single cell.
Great. If there's no more questions, let's thank our speaker again. Thank you, Mary.